where I'm very fortunate to be involved in our CCTS as Foundation Director for Precision Medicine. And with Paul Bray, I'm also very keenly interested in platelet genetics, so I was really excited to hear the first two talks, which were spectacular. I'm pleased to introduce our next session, titled Precision Medicine and Population Health in Action, Cancer Genetics. I'll briefly introduce the first speaker, and then after that, the second speaker. The first speaker is Dr. Jen Doherty, who is an associate professor at the University of Utah. She's in the Population Health Sciences and Cancer Population Science departments. And her work is focused on identifying the epidemiological, genetic, and molecular factors that influence outcomes in ovarian cancer. And without further ado, Dr. Doherty. Thank you.
Thank you. I'm really thrilled to be here. Let's see. So today, my story, it's a very short story, because I only have 10 minutes, but it's about how important it is to define phenotypes in something like a disease like ovarian cancer. And how does this impact population health? Ovarian cancer is extremely rare. It accounts for only 3% of malignancies, but it's the 5th leading cause of cancer death in women, so it's quite fatal. The reason for this is that it's usually diagnosed when it's spread throughout the peritoneal cavity and diagnosed at a late stage. 5-year relative survival is 46%, but it's considerably worse at late stage. Treatment consists of removing the tumor from the peritoneal cavity and platinum Taxol combination therapy, which hasn't been very effective over time, actually. Neoadjuvant therapy is common in some places, but it's controversial. And there's a lot of interest in doing immunotherapy in this disease. So survival is dependent on the amount of disease at diagnosis, but then how much of the disease is able to be removed and what's remaining. Also, histotype is extremely important, as I'll show you soon. So traditionally, ovarian cancer has been diagnosed using morphologic features. But over time, people have understood that protein expression varies widely across different ovarian cancer types. And so these are the morphologic types, and these are several protein markers. And you can see dramatic differences in the expression patterns across the morphologic types. So a group that we actually work quite closely with has developed calculators incorporating these protein markers and grade. And all of this work over many years has culminated in the understanding that we've really been misclassifying phenotypes of ovarian cancer pretty badly. So the new classification has this distribution versus the old distribution of histotypes. At the same time, it's understood that despite for 40 years believing that the cell of origin for ovarian cancer is the ovarian surface epithelium, most high-grade serous ovarian cancer arises from cells in the fallopian tube that migrate. Also, for the endometrioid and clear cell subtypes, the cell of origin is endometrial tissue. So there's a broad consensus and understanding that ovarian cancer is not a single disease, but it represents a cluster of diseases that all happen to occur around the ovary and actually the peritoneal cavity. To give you a sense of the impact of these changes in our understanding, one of my colleagues looked at a set of tumors that he had classified using an old classification in 2002. He applied the new guidelines to the exact same set of slides. So all of the slides were the same, and came up with the 2014 classification here. He also asked a colleague that he didn't actually know very well from a different city. He sent all of the same slides to his colleague and asked him to also use the 2014 guidelines to classify the tumors. Remarkably, this pathologist was not concordant with himself before and after these guidelines, but he was nearly entirely concordant with his colleague using these new guidelines. So this has profound implications for how we think about ovarian cancer. Basically, all of our studies in the past are misclassified. The problem with ovarian cancer is it's extremely rare, and so we can't just wait for cases to accrue. We need to figure out ways to apply our understanding now to what's happened in the past. So what we have done in my group is we got together with our pathologist colleagues who've been involved in developing these guidelines, and we sat down with them and with the SEER data, this is about 28,000 ovarian cancer cases, and reclassified the ICD-10 codes as best we could. We don't have protein markers, but knowing what we know now, we were able to reclassify the codes, and also we used data on grade, and we looked at survival by stage. And if you read the ovarian cancer literature, you'll see again and again that people say that high-grade serous ovarian cancer is the most lethal form. In fact, what we see now that we've disaggregated the phenotypes appropriately, we see that actually mucinous and clear cell types, the blue and orange here, have dramatically worse survival when diagnosed at late stage. Carcinosarcomas, these are the red line. We've known that those have the poorest survival regardless of stage. But high-grade serous is the black line here, and it certainly does not have, it is not the most fatal. So anything that we do moving forward, we need to apply this new understanding of these patterns of survival, and we need to take histotype and stage into account in all of our analyses. Again, this is a very rare cancer, so it gets dicey with numbers. TCGA, along with many other cancer types, has developed subtypes of high-grade serous ovarian cancer. And this is something that's been replicated in a few different populations. And there's reason to believe that there might be biological differences between these gene expression subtypes. There are differences in survival. There are differences in the ability to debulk these tumors or remove the tumor. And then also there are differences in expression of immune cells across these types. So this is a very exciting development. There's also, to apply this to precision medicine, post hoc colleagues went back and typed the tumors that were in this bevacizumab trial and observed differences in median improvement in progression-free survival that were quite different across the gene expression subtypes. On top of all of this, it's well known that ovarian cancer is genomically chaotic because it's characterized by extensive copy number alterations compared to other cancer types. Also, mutations are essentially private. Other than P53, the recurrent mutations, there are very few recurrent mutations, and methylation is also quite quiet. So in my group, we've put together teams of multidisciplinary scientists to try to understand etiologic factors. So this is where epidemiology comes in. For both protein expression defined histotypes and also the high-grade serous gene expression subtypes. And my ultimate goal here is to develop an understanding so that preventive strategies can be developed. We're also trying to refine our understanding even more for the histotypes and gene expression subtypes so that we can identify molecular features and pass the baton along to people who understand how to develop targeted treatments. So our work in refining the subtypes has really consisted of doing these cross-population comparisons to understand how robust subtypes are across populations. I'm not going to spend a lot of time describing this today, but I do want you to know that this can be applied to any disease type, so if you're interested, come talk to me afterwards. The other thing that we're doing is, again, in order to leverage the existing data that we have on ovarian cancer, we're trying to develop a reduced gene set classifier similar to OncoDX, which was mentioned earlier, or PAM50, which is something that was developed here. And the idea is that we could use a simpler test to go back, understand what's going on in our existing data, but also move forward in clinical studies and our new enrollments. In order to do this, though, in ovarian cancer, it's taking a collaboration with 20 other investigators, and we're only looking at about 5,400 tumors, which is huge in ovarian cancer, but I know anybody who does breast or prostate, it's not that many. But another application that I'm particularly excited about in refining these phenotypes is to understand whether they might explain some of the differences that we see in ovarian cancer survival across races. And so African American women have much worse survival from ovarian cancer than do women of European ancestry. So we asked whether African American women, whether we can observe the same subtypes across African ancestry and European ancestry women, and also whether African ancestry women may have more aggressive subtypes. This is a heat map showing you the TCGA subtypes in European ancestry women and African ancestry women. Red indicates high correlation between differential gene expression across 10,000 genes in each of these. And what we see is that there's very high concordance between two of the subtypes. These are actually the more aggressive subtypes. We also see moderate concordance with a third TCGA-defined subtype. But what's remarkable to us is that we find a fourth subtype in African ancestry women that we can't map back to any other population, including Asian populations. So this is somewhat pilot phase data. It's on about 120 African American women. And we're doubling the sample size later. But anyway, I just want to point out that this unknown subtype makes up more than a quarter of the African ancestry cases that we have. But also, indeed, we are seeing that this more aggressive subtype in blue here does appear to be more prevalent among women of African ancestry. So moving this forward to really understand what these subtypes mean, we're taking 250 African ancestry women, typing their tumors not only in terms of gene expression, but layering essentially every omics measure possible. And what we're doing is using pathway-based approaches to identify modules of interrelated pathways within these subtypes. And then I mentioned that ovarian cancer is chaotic genomically, and we haven't been able to use traditional tools to understand what's going on in terms of drivers. So we're further applying our methods. We're identifying pathways in both European ancestry and African ancestry women, and then overlaying them and trying to find where we see overlap. And so these areas become the top choices for functional follow-up. So to summarize, sorry it was such a whirlwind. 10 minutes is actually pretty hard. So changes in the understanding of phenotype have incredibly large implications for prevention and treatment of disease. But also, the other point that's true for ovarian cancer is that genomic complexity is something that requires new approaches. And so we're trying to marry our both epidemiology and population health sciences with bioinformatics, pathology, and other disciplines in order to get beyond this impasse. With that, I'd like to acknowledge a short list. There are many more people, but anyway, so I can take questions if you'd like. Okay.
Thank you very much. I think we have time for one question.
So I have a question about phenotyping, because I think that it's very interesting that when you take some features of a phenotype, you may not have good agreement across people that are assigning that phenotype. And so was the switch from the ovarian cancer classification scheme, was that a pathologic base, and was it, so clearly it wasn't the pathologist, it was the phenotype classification. And could you comment a little bit about what was different going from 2002 to 2014?
So what the way we are, the label that we use to define what happened is that it's IHC augmented expert pathology review. So the core is still the expert pathology review in terms of morphology, but there have been big changes in terms of understanding, for example, P53 and WT1 are really defining features of high-grade serous ovarian cancer. And if you don't have those, then it shouldn't be defined as high-grade serous. But if you do have those, that's where, high-grade serous went from about 60% prevalence to 75% prevalence. So that really changed a lot. The other big change was the understanding that grade helps distinguish endometrioid tumors. So low-grade endometrioid tumors are very different from high-grade endometrioid tumors, but most high-grade endometrioid tumors are actually molecularly more similar to high-grade serous. So yeah, it's pretty detailed, but it does have to do with a combination of morphology, molecular testing, and understanding of the impact of grade.
And clearly a lot of training for the pathologists.
My pathologists always say, they're Canadians, so they're really funny, but they always say that a trained monkey could do it. So I'm not sure. I think they're beyond most monkeys I've seen, but there are a few. So for the majority of cases, it's easy, but there are always these controversial ones, and there remains quite a large controversy about low-grade serous. We need to refine that phenotype even more, but it's quite rare, so it's hard. It's even harder to look at.
Okay, thank you very much. Our next speaker for this session is Dr. Kathy Cooney. Dr. Cooney is professor and chair at the Department of Internal Medicine, and her research work is focused on the genetics of prostate cancer, including identifying gene variants associated with increased risk of prostate cancer and studies to improve the utility of prostate cancer in higher risk men. Welcome, Dr. Cooney.
Thank you, Matt, and it's a pleasure to be here today in my home. Thank you to the organizers, and thank you for the challenge of presenting in 10 minutes, very complex topics. And so, as Matt said, I am a medical oncologist. I take care of men with advanced prostate cancer, but most of my research has been in genetic epidemiology, and it's been a very challenging 20, 30 years trying to identify gene mutations that have clinical relevance. What's happened over the past about 5 years is work in genetic epidemiology has sort of intersected with precision medicine to lead to some really important and clinically relevant observations, and that's the story I'm going to try to tell in 10 minutes. What you can see is that prostate cancer is the most common cancer diagnosed in American men, excluding non-melanoma skin cancers. This number has declined somewhat, and we'll talk about why that's true. And I think the other important thing to, as a clinician, we often hear that people die with prostate cancer, not of prostate cancer. And it's important to remember that prostate cancer still is the second leading cause of cancer deaths in the U.S. So I think important data. So what's happened, though, over the past 30 years of a very complicated, long story is that in the early 1990s, PSA testing was introduced into the population with lots of promise. So PSA is basically it's a serine protease. It's made by the cells of the prostate gland and also by cancers that arise from the prostate gland. And what happened, again, summarizing a very long story, that between the 90s and the early 2000s, there was a dramatic increase in the number of prostate cancer cases diagnosed. And in retrospect, many of these cases were low-grade and low-stage, the kind of things that my husband's a surgeon, urologists love to treat because patients do well. But in reality, lots of epidemiology studies have suggested that many of these cases probably did not need to be diagnosed and treated, and has created a really difficult time in our field. What's happened now in terms of the public health impact is that there's been quite a number of changing recommendations about the use of PSA testing in the population. So in 2012, the U.S. Preventative Services Task Force decided that they would change the recommendation and make prostate cancer screening a D recommendation. In other words, the harm created by either finding cancers and treating cancers that maybe didn't need to be treated and exposing men to a lot of maybe tests to work up elevated PSAs in patients who didn't have cancer, sort of those risks outweighed the benefits of finding cancer earlier, treating and maybe curing individuals. This year, we expect that this recommendation will turn to a C recommendation. It has not been finalized, but the C recommendation means for clinicians and patients, there's a balance. The doctor and the patient need to sit down and talk about the risks and benefits of prostate cancer screening in that individual. So again, while all of the change in how we manage prostate cancer is taking place, many of us were out there trying to understand the genetic epidemiology of the disease, and it's been pretty complicated. So the key questions I think I want you to think about in the next 8 minutes is, can we use genetic markers to help identify men who will develop prostate cancer? Can this be supplement to PSA or other types of screening for the disease? But more importantly, can we use genetic markers to find lethal cases of prostate cancer, really hone down on those parts of the disease that actually impact life expectancy in men? So again, summarizing lots and lots of data, lots of work that I've done with a number of colleagues throughout the United States and the world, there's been a lot of focus on finding rare variants in prostate cancer. We had heard Dr. Croydon talk about maybe not enjoying the concept of heritability, but we do know that prostate cancer clusters within families, and it suggests that some of this may be epidemiology, so other environmental things, but certainly genetics probably plays a component in explaining some of the clustering of prostate cancer cases in families. So again, summarizing is just lots and lots of literature. Really the most important observations during this time is that it was observed that in families with hereditary breast and ovarian cancer, male mutation carriers, particularly BRCA2 mutation carriers, have a higher risk of developing prostate cancer. But the penetrance of for example, BRCA2 mutations for prostate cancer is not as high as in breast and ovarian cancer. So you don't generally see in the clusters of hereditary and breast and ovarian cancers as many cases of prostate cancer, nor did we find families with multiple cases of prostate cancer and BRCA1 and BRCA2 mutations. Lots and lots of work studying lots and lots of families. The only gene that's been discovered that is a prostate cancer specific susceptibility gene was discovered by my laboratory in collaboration with Bill Isaacs at Johns Hopkins and a number of other teams throughout the United States. What we discovered doing linkage studies, so this is again the only gene found by linkage studies, is that there's a mutation in the HOXB13 gene, which is on chromosome 17, and it's a recurrent mutation. Again, a very interesting story in cancer susceptibility. It's a G84E mutation seen in almost every single family that has prostate cancer due to this gene. It accounts for about 5% of hereditary prostate cancer worldwide. This recurrent mutation is on a common haplotype, which means it's likely a founder mutation arising from Nordic populations. But despite lots and lots of family-based studies, we were unable to find a high number of rare mutations in genes that might cause prostate cancer. But what I want you to remember here is that we were looking at a time where lots of indolent cases of prostate cancer were diagnosed and likely diluted the samples that we were studying. So, move forward now to about 2010, there was this sort of, large movement toward sequencing tumors, with the idea being that we could find mutations in tumors that would help direct therapy. And so I spent much of my career at the University of Michigan, worked with lots of really wonderful people, in particular Arul Chanayan, who leads the prostate cancer spore there at Michigan, and is also the lead of the Stand Up to Cancer grant on the East Coast. And so we had the opportunity, because of these large grants, to sequence tumors from many, many prostate cancer patients. So this is an example of that type of approach. I think I showed this slide last year, which is a case of a patient. This is a real patient from our clinic, 55-year-old man, which is a young age at diagnosis for prostate cancer, presenting with very advanced disease, node positive metastatic disease. This is an actual bone scan showing the high degree of sclerotic metastases that this patient had at the time of presentation. What was done was, instead of biopsying the bone, which often doesn't reveal a lot of cancer cells, we actually biopsied a metastatic lymph node. As you can see, this tumor, for pathologists in the audience, is a very aggressive, high Gleason grade tumor. What was found on tumor sequencing was the patient had a TMPRSS2 ERG fusion, which is very common, seen in about 50% of prostate cancers, but interestingly had both a somatic and a germline BRCA2 mutation, so a double hit. And interesting, when you go back and talk to this individual, his family is very typical for a breast and ovarian cancer family, multiple cases of breast and ovarian cancer, which I think maybe wasn't fully appreciated at the time that this individual presented. And if you want to, this is a very nice reference from Samikra Chowdhury, who now runs the Precision Medicine Group at Ohio State, describing sort of overall how this type of approach works. So between 2010 and about 2016, many, many studies like this, looking at men with advanced prostate cancer, biopsying tumors, and looking at what types of mutations were discovered. And again, trying to summarize a large body of literature. The major two citations at the bottom summarize data from about 800 or 900 men with advanced prostate cancer who underwent tumor sequencing. Bottom line is that if you look at all of this body of work together, it appears that there are germline mutations in a panel of DNA repair genes in about 10% of patients. And this graph that I'm showing here is from the Pritchard paper, Colin Pritchard at the University of Washington, and shows the distribution of where these the gene panel that these deleterious mutations occur. The bulk of the mutations occurring in BRCA2, and then the other major players, ATM, CHEK2, and BRCA1. And what's quite striking is that, these are all DNA repair genes. They work in the same pathway. And again, confluence of interesting observations. There were a number of PARP inhibition trials done at the same time, and it shows that men who have mutations either somatically or germline in any of these panel of genes preferentially respond to this medication. So this knowledge is not just important for the families and understanding heritability, but actually has implications for treatment. So what does this mean at a population level? This is a paper I was fortunate to participate in with my colleagues again at Johns Hopkins and North Shore, where we were able to go back and do a retrospective analysis then of lethal cases and indolent cases derived from the same population, so a case control study. And what we were able to show is that BRCA1, 2, and ATM mutations were more common in the lethal cases compared to the indolent cases. So an intersection now of genetic epidemiology and sort of knowledge that we've learned by being in the clinic and studying men with cancer. And additional studies show that mutation carriers are more likely to die from the disease, and they're also more likely to die at an earlier age. So I think very important data. So a number of us got together, gosh, it's been almost a year ago, and met at Jefferson, and it was a really interesting meeting of oncologists, geneticists, urologists, family practice doctors, and tried to figure out what should we do with this, this information in the absence of really data that says that screening is really beneficial, finding these diseases early and treating them is going to, is going to cure people. But we felt compelled to start to make some recommendations, and so a lot of this is not evidence-driven, but panel of expert recommendations. And this is actually just came out about 2 or 3 weeks ago from the National cancer, it's the NCCN, which is a group of cancer centers that make recommendations for oncologists and clinicians, and they've really increased the recommendations and suggest that all men with high risk or very high risk or advanced disease should be tested for germline mutations, particularly the panel of DNA repair gene mutations as well as HOXB13, and that additionally men who are in families that appear to have syndromes, hereditary breast and ovarian cancer, perhaps Lynch syndrome, should also be considered for genetic testing. So this is really going to increase the interest in genetic testing to a large sector of the population, men in general, who have not participated in this type of studies previously. So again, working together with a number of groups, we have just opened our prostate cancer risk clinic here at Huntsman, which is a collaboration between Sam Greenberg, I see her in the back, Brock O'Neill, one of our urologists, and myself. Will be working with men who have been identified to have BRCA1, BRCA2, or other mutations, figure out what the right plan is for them. Is it yearly screening? What's the role of PSA? And we've been fortunate to connect with a number of other groups doing similar work, some at Michigan, at Fred Hutch, so we can collect data sets that are together, because again, these are uncommon cases, uncommon individuals, so we need to work together to put together cohorts where we can get meaningful data. So not a conclusion, but a summary, that rare germline variants in cancer predisposition genes do increase the risk of prostate cancer. For some DNA repair gene mutations, we think there's an increased risk of lethal prostate cancer. We've been able to prove that, I think, pretty compellingly for BRCA1, 2, and ATM. It's not clear for the other genes that are more rare. And we really need new strategies for early detection, and we need to bring in other types of testing other than PSA testing to help find prostate cancers and treat the cancers that are likely to kill men. And with that, I thank you and be happy to take questions.
I think we have time for one question.
Wow.
I might take the liberty then. I was interested in whether you've identified or others have identified differences with regards to these mutations by race. We heard earlier from Dr. Doherty that race can influence ovarian cancer. I was wondering if there's similar data in prostate cancer.
Well, I think I see Rick in the audience. I think he may be discussing this at some point. I don't know, but the data. We know that African American men experience prostate cancer at a higher rate. They have more aggressive disease and more likely to have lethal cancers. There have been many studies trying to figure out if there's a genetic component to that. I think that the answer is really not clear at this point. We actually do have. I have a Department of Defense grant with one of my former trainees at the Detroit SEER, actually enrolling a number of patients diagnosed with prostate cancer, African Americans only, before the age of 56, to try to start to identify what the frequency of germline mutations are in this cohort. And then my colleague Jen Beebe-Dimmer also got a second DoD grant to look at families with clusters of breast and ovarian cancer to see if we can also define, how much BRCA1 and 2 mutations are in this population, and are there other genes that account for not just prostate cancer, but prostate and breast cancer in African Americans. So lots of research that I think now can move forward, but is challenging, often due to the fact that we don't recruit enough African American and minority patients into our cancer studies.
Thank you.
Thanks.
So I want to thank Dr. Doherty and Dr. Cooney for those wonderful talks. We're going to take a 10-minute break. We'll like to reconvene at 10:45 for our next session, Precision Health, Population Health, and Health Disparities. Thank you.